few heavy hours of N and O glycans and sialic acid based glycans, we have something really light, which is the reason why I don't have handouts for all of you. Obviously the trainees have in their pack, and I really want you all to simply focus on the fundamentals, really understand chromatography from the perspective of how interactions occur, how resolution occurs, and be in a position to exploit these simple rules. Okay, so this is going to be a short talk. At the end, I have a paper for you that I have made copies of, which I'm going to distribute to all of you. And basically, we are going to read the paper. It's a chromatography-based paper that has some nice information, very recently published. A very simple paper that you all should be able to read right here and be able to interpret and get the essence of using chromatography to purify, to resolve, to isolate glycans. Okay, so in general, we can classify glycans into two major categories, two major types. Type 1 are the neutral glycans, okay, the neutral glycans, and type 2 are the highly sulfated glycans. So the neutral glycans are, for example, what we studied just now, the N and O glycans. The neutral glycans may or may not have sialic acid, and sialic acid is charged, right? Under normal conditions, the carboxylic acid of sialic acid is charged, and yet that is still classified as a neutral glycan. Okay, and the heavily sulfated glycans are obviously heavily charged. So these are the two major classes, and much of what chromatography is about understanding the structure of glycans so that we can choose an appropriate technique, for resolving and for purifying these glycans. So if you really look at what techniques have been developed from the point of view of chromatography, this is what we have. We have several techniques that have been utilized. Some of these techniques are really very useful. They are very powerful, and we can understand a lot about the structure also based on chromatography. So for example, we have affinity chromatography, we have ion exchange chromatography. Ion exchange can be of two types, an ion exchange or cation exchange. We have hydrophilic interaction chromatography, which is called as HILIC, H-I-L-I-C. We have reversed phase chromatography. We have a usual reversed phase, or we have reversed phase ion pairing called RPIP chromatography. We have size exclusion chromatography, and I also brought in electrophoresis. Number of times electrophoresis is classified independently, but I simply brought it in primarily because it is also a resolving technique, and number of times under really extreme conditions you can also use electrophoresis for preparative purposes, just as you can use electrophoresis for identification, analysis, and those kinds of things. A major part of chromatography of carbohydrates revolves around the difficulty of detection. As we saw yesterday, and as Kubi presented yesterday, the chromophores, or the tools that are really available with regard to glycans as well as sulfated glycans in terms of detection are not very many, right? If you really look at the structure is primarily, you have these hydroxy groups, you have an aldehyde, sometimes you have a carboxylic acid. So in essence, how are you going to detect? Does anybody know as to where a hydroxyl group, anything particular about hydroxy group that can be used for detection?
IR spectrum.
Yeah, you can use an IR spectrum, absolutely. It has a specific, if it is hydrogen bonded hydroxy or if it is non-hydrogen bonded hydroxy, you can use an IR technique to detect an OH group. With regard to chromatography, IR as a detection tool has not really been exploited much. It's not really a method that people use. Why is that? With respect, especially with respect to glycans. Obvious answer. So most of the time, obvious answer. I'm absolutely certain you know. What's that? There are multiple hydroxy groups. So if there are multiple hydroxy groups, detection using IR should be more facilitated because you have a higher extinction coefficient. And yet IR, glycans, is not typically used. Hydroxy. Why? Obvious answer. You don't have to think a whole lot. Degenerate.
All the most of the OH groups are similar to
Oh, okay. Yeah. So if you are thinking along the lines that all the OH groups are similar, so all carbohydrates will essentially give you similar peak. But then when you do chromatography, you are hopefully going to resolve different carbohydrates. So you should be getting different peaks. Okay, let me give you a clue. Carbohydrates are soluble in water. So you are going to have a phenomenal water background, typically, right? So it doesn't really work. What about, somebody asked a question.
Can't you dissolve them in some other, like DMS or some other solvent?
Yeah, you can dissolve partially. You can dissolve partially in very highly polar, but then that really limits the resolution power of the technique. Okay. What about other functional groups in carbohydrates that you know of? What are the other functional groups? Aldehyde. Let's say there are aldehydes, assuming that you do chromatography of monosaccharides, for example. And how many times are you going to do that? But sure. What about it? Can we use a direct detection technique for aldehyde? Once again, IR, right? I don't know whether you know, but aldehydes or carbonyls have a specific peak. Sure, extinction coefficient is not that high. So even aldehydes is not typically useful. Carboxylic acid, very similar problem. So what happens is that carbohydrates are difficult to detect directly. Carbohydrates are difficult to really identify directly. So therefore, detection becomes a major issue with regard to carbohydrates. And therefore, several techniques have been engineered to help with the detection. One of the techniques, obviously, is to introduce a chromophore or a fluorophore. Right? What is a chromophore? A chromophore is a group that, is a UV active group, UV visible active group. We typically call it chromophore. What about fluorophore? It's a group that exhibits fluorescence. So a number of times these chromophores, or for primarily chromophore, can be intrinsic. And we are going to see one example. Kubi has already talked about that series of molecules. Can be intrinsic. So if it is intrinsic, obviously that helps detection. But majority of times, chromophores have to be tagged. Chromophores have to be attached. So there is usually an attachment step, a chemical step that is involved to help with detection. A major technique that has been developed, that was developed, which these days is not much utilized, although you can see papers on this, is called pulsed amperometry. Okay, this is where oxidation of oxidation of carbohydrates is engineered, and that generates a current and is detected. And the one technique that has now developed to such an extent that we not only use this for detecting carbohydrates, but we also use it for identifying. Specific identification of structures can really be achieved with mass spectrometry. And these days, we always use mass spectrometry in combination with chromatography. So what I'm going to talk about primarily is liquid chromatography based carbohydrate resolution, detection, and identification, where MS forms a significant component. Okay. All right. So what we are going to do in the next, let's say, hour or so is to simply understand the principles of these techniques. And I have a very succinct figure that I want you all to simply focus upon and basically understand the different techniques at a very fundamental level, and hopefully you won't forget it. So here are three techniques. We'll start with affinity chromatography. Okay, so we'll start with affinity chromatography. So affinity chromatography essentially relies on the affinity of two molecules. One of them obviously is the glycan. Okay, the other can be whatever the interacting partner is. And in essence, what is done is that the matrix that we utilized in the column is loaded with one of the partners. It can be a receptor, it can be lectin, it can also be the glycan that we are trying to measure. In that case, the partner becomes the lectin or the receptor or other interacting protein. So out of many molecules that might be present in the sample, we design conditions such that we introduce favorable interaction. And once those conditions are designed, then we expect that the molecules that are present in the sample, a few of them would actually bind with the matrix which has been loaded with an appropriate molecule, appropriate receptor, or lectin for that matter. So as far as resolving carbohydrates is concerned, imagine that the sample is simply a mixture of carbohydrates, and what we have loaded is a protein of interest to which we expect one or more of the carbohydrate to bind. So under appropriate conditions, these would bind, and then when you elute with a buffer containing an appropriate competing ligand or a change in the condition, it could be a pH change where the interaction is disrupted. It could be a change in the salt concentration. It could be a change in other conditions that might disrupt the interaction. What typically happens is that you release the one that actually binds, the last arises with a relatively small peak, which is the high affinity molecule that binds the receptor. The rest of the fractions that do not bind the receptor simply flow out in the initial buffer. And that indicates that this particular molecule is the one that has high affinity for the receptor that you have chosen. It's a very simple technique. It relies on the idea that this particular molecule has a higher affinity in comparison to the rest of the molecules. It doesn't have to be the highest affinity. It can be a marginal increase in affinity also, and you would be able to see resolution. So you would get this particular sequence coming out much later. This particular sequence can then be detected, can then be isolated, can then be purified, and then quantified using different techniques so that we can characterize this. Mass spectrometry obviously typically is very useful in this. Now if there are several types of molecules. For example, as we have seen that mannose is a component in several N-glycans, or galactose is a component in several N-glycans. Some have sialic acid, some don't have sialic acid. You might have varying number of sialic acid residues. Then the affinity would change. So you would then have multiple peaks that might arise. And then the affinity would then determine as to how the resolution is. So instead of having just one, you might have several. And then depending upon the affinity, the one that has higher affinity would turn out to be the last. So it's one easy way of even identifying the relative affinity. You won't be able to identify affinity in a quantitative manner, but you'll be able to identify the relative affinity of these molecules for the lectin. Imagine, expand it even further. If, for example, you have this matrix made up of several different receptors, several different lectins, you can actually have an array and use affinity to identify the specificity of the lectins or a series of lectins. Okay, so this is a fairly powerful tool for purification purposes, and is one of the ones that has high resolution, and one of the ones wherein even if you have 1,000-fold greater amount of the non-binding component, you will still be able to identify this. Very, very few techniques would be able to do it. Okay, that you would be able to pick out even minor components. All right, clear? Let's move on. Affinity is something that is very simple to understand, and I think affinity is something that many of you, how many have performed affinity experiments? Okay, quite a few. So, all right. So, let's move on to size exclusion. Size exclusion chromatography is, in essence, exactly as the name says. Size is excluded. Okay, what is size here? Size is essentially molecular weight. It's not really molecular weight, but it's the overall volume, the hydrodynamic volume. Because carbohydrates have number of OH groups and they tend to be associated with a lot of water. Sulfated carbohydrates tend to be associated with even higher proportion of water. So, depending on the size of the molecule, certain molecules will be excluded and they would come out at an appropriate time. So we are, in essence, using a technique of separating molecules for most part according to size. And this relationship can really be exploited to determine the molecular weight. That is the interesting part, because carbohydrates are typically polymers. Carbohydrates, and for polymers, the biophysical property, one of the biophysical property is the molecular weight. Not only just molecular weight, is the number average molecular weight, or the weight average molecular weight, or the peak average molecular weight, or the viscosity average molecular weight. So there are several molecular weights as far as polymers are concerned. And this is a technique that is typically used most commonly. So what is done is, obviously you have a mixture of molecules, let's say of different sizes as indicated by these spheres. You have a matrix. This matrix is typically porous. This matrix is porous and is able to separate molecules according to size. Certain molecules will be able to penetrate the matrix. Certain molecules will not be able to penetrate the matrix really well. So because of these differential penetration, those molecules that are able to penetrate the porous nature of the matrix would actually go in, and those that cannot penetrate would actually bypass the entire the channels that are formed in the matrix. So smaller molecules typically are retained more than the larger molecules, and eventually what typically happens is that in the first few fractions you would see much larger molecules, followed by smaller molecules, followed by even smaller molecules. So this is exactly opposite of what conventional thinking is. Conventional thinking would be that for small molecules, the number of interactions, the number of OH groups, the number of things that could actually interact are less, and therefore would not be retained as much. Here, it's exactly opposite. Bigger molecules would come out faster. Smaller molecules would come out slower. The retention time here can be easily correlated with the hydrodynamic volume and therefore the size of the molecule. Okay? And in both of these, typically the eluent is essentially aqueous base. So you have all water-based system. The pH that can be chosen in both of these can be as close to the pH of the optimal pH of the system that you're working with. And therefore both of these techniques are very useful techniques. Okay? Any questions? Yeah.
How does the shape of the molecules affect the dilution time? Like, for example, especially for the polymers, some have like linear chain and some have branches. So could this method separate those polymers that have different shapes?
Absolutely. Yeah. It's not just the molecular weight. It's not obviously just the hydrodynamic volume, but also the shape. On the extremes, if you have a globular carbohydrate versus you have an absolutely linear carbohydrate, there is a phenomena called rectilinear motion. So it works as a reptile and is able to, some of the molecules might actually be able to enter the pores and may be retained. So yes, you would have some phenomena wherein the shape also determines. But I'm going to show you a profile where the relationship between the migration time and the molecular weight is not a linear relationship. Okay? The relationship is a semi-log relationship. So when you have a semi-log relationship, the molecular weight change has to be significantly more in order to affect a small change in the retention time. So when we are comparing two polymers, one that is absolutely globular and an equivalent weight which is absolutely linear, then you will see some differences, but you will not see phenomenal differences. So it will not be a complete turnaround. Okay? You do have to utilize controls. Not only that, you have to utilize appropriate controls. Just as we saw yesterday in carbazole assay, you can't really use completely different structural controls in carbazole. You have to use an appropriate control. If you are trying to quantify uronic acid in heparin, then for most part you have to use a heparin-based control or a reasonably close control. Then the correlation becomes much higher. Very similar. Here, if we are using, let's say, if you are doing molecular weight determination of low molecular weight heparins, I think I have a slide that's coming up pretty soon. And low molecular weight heparins are heparins. They are heavily charged molecules. And we try to use controls that are based on dextran, unsulfated dextran. So you're comparing unsulfated dextran with sulfated heparin, then the correlation will not be very good. Okay? The correlation will, there will be a systematic drift, and therefore you might be way off. So you had to use an appropriate, you can use dextran, but then that has to be sulfated. Okay? Because the hydrodynamic volume is going to be significantly different. The number of water molecules that each sulfate group carries is going to be significantly different. Just for your information, every sulfate group would approximately carry 9 water molecules. So think about it. Okay? Every sulfate group would approximately carry 9 water molecules, approximately, okay? Give or take one, one or two. So we are looking at, if a sulfate has a molecular weight of 102, okay? 98. With sodium. With sodium. SO3 is 80. With sodium, it's going to be 103. Okay? Yeah, SO3 is 80, plus sodium is 23. minus 1, so it's going to be 102. So 102 and 9 water molecules, so 9 times 18. 20 times 18 is about 180. So you can see one sulfate group has an equivalent additional molecular weight approximately. So that's a huge impact if you figure it out over all of it. Okay, can I go forward? Yeah? All right, let's talk about ion exchange. Okay, so one of the things that I completely forgot about is here the matrix plays in essence no role other than sieving. The matrix doesn't play a role, and it would be difficult to find a matrix that has absolutely no interaction with molecules. Okay, you would have some interaction. So when you start comparing different matrices and assume that certain person has done this analysis with this particular matrix and I'm going to do with this matrix and extrapolate the results, that might not happen. Every matrix has to be calibrated with an appropriate standard. And you have to be careful. There are some matrices, polyacrylamide based sieving matrix. Polyacrylamide typically interacts, good hydrogen bonding capability, good hydrophobic interaction. So you have to be careful. Okay, ion exchange. Ion exchange chromatography, and herein is a figure that is primarily for what is coming out first. So this is positive, this is negative. So this is anion exchange chromatography. Okay, so this figure is primarily for anion exchange, but obviously it can work for cation exchange. So in ion exchange, we have a matrix that has either positive or negative charge. Okay, and the idea is that if the sample has a charge, then the opposite charge would be attracted, same charge would be repelled, and the molecules that have same charge would come out first. Or in this particular case, there are 3 molecules, one with only 1 negative charge, 2 with 2 negative charges, and 3 with 3 negative charges. So depending on the number of charges, the interaction probability is going to be much, much better. So under appropriate conditions, typically under low salt conditions. So ion exchange is primarily a phenomena wherein if you have low salt, you would not be screening these interactions, the charge interactions, and therefore you would encourage the interaction. So under low salt conditions, you would have these interactions more favorable than these as compared to the unitary charge interactions. And as you increase the concentration of salt in the elution buffer, as indicated by here, so as you increase the concentration of salt, you would see that the lower affinity molecules would come out first, followed by the intermediate, followed by the highest affinity molecules. So salt is a dominant feature out here. Obviously the ones that have no charge would come out primarily would not interact at all, and you would see something like that. So you would have, if nothing interacts, you would have a fairly big peak. Most of the time that happens with heparin when we do heparin cephalose chromatography. At the end, those molecules that actually bind the best typically elute at a fairly high salt concentration. So what happens? In your eluent, you would get high salt. In your eluent, you would get fairly high salt. So then you'll have to desalt in order to really purify. And desalting is a challenge. Desalting is a challenge. Why? Especially for glycans. Once again, simple things that you know of, but I want you to think. By the way, we are going to do chromatography, right? Today. So, why is desalting a problem? It precipitates. If you reach that high concentration, sure, it precipitates. Let's say that you have, all right, I'll give you a clue. I'll give you an example. So typically what happens is that you start with, let's say, 20 millimolar salt. Very few times you start with zero salt. You can, but under zero salt conditions, not very helpful in terms of lots of other things. You start with a very small amount of salt and you gradually increase it. And really high affinity, I'll give you an example. So antithrombin binding to heparin is a very tight interaction. Okay, the affinity for polymeric heparin is sub 10 nanomolar. It could be 4 nanomolar, 5 nanomolar. It's fairly strong. You disrupt that interaction between antithrombin and heparin, for example, with very high salt. Now, obviously, you don't use antithrombin here. In affinity, you use antithrombin in order to purify. But with ion exchange, where you have this matrix, where you have a lot of positive charges called strong anion exchange, even there, the affinity of heparin is really, really high. So you end up with close to 1 to 2 molar salt concentration, which will be able to release heparin, right? So 1 to 2 molar salt, and typically what happens is, unless you are purifying microgram quantities, if you do several milligram quantities, then you end up with fairly high concentration of salt, 1 molar salt or 1.5 molar salt. And if you have oligosaccharide, not heparin, but if you have oligosaccharide, then that is a significant challenge from the preparative perspective, which I want all of you to understand. And the question is why? Okay, go to that setup. If you go to the setup, you will realize, what is going on. Sorry, somebody said something. Spencer?
If you have to get rid of this all, then we can do dialysis.
Okay, that's a good point. You can, obviously you have to do dialysis. These days there are much better tools than doing simply the regular old style dialysis that we used to do, especially in the long tubes. You put it in a buffer, 2 liters of water, keep floating. We have much better tools. Karthik is laughing because he's done it several times. Oh, you are doing it today, okay. Okay. But, yeah, okay. So for those of you who don't really know how for example, dialysis works is, what happened? Dialysis works is that I don't have a figure. But in essence, what do? You basically, a simplistic diagram would be that you have chamber 1, and there is usually a membrane, and there is chamber 2. Okay? So, and this membrane has molecular size pores. So this is not a filtration membrane where you filter particles. Okay? This is actually molecular size pores. And these pores determine what molecules can actually pass through from one chamber to the next. Okay? So this could be a 2-chamber system, or it could be that you have this membrane itself forming a chamber which you put in a flask, and all around there is water. So this is your water or buffer or whatever that is. And this is your molecule or glycan containing solution. So you would have all the unnecessary or whatever small molecular weight that you really want to lose come out from here. So this is how typically chambers work. Dialysis or separation works in the molecular weight system. These membranes have a molecular weight cutoff. For example, these membranes have a molecular weight cutoff number listed. For example, something called 1,000 or 5,000 or 10,000, and likewise. So every molecule that is less than 1,000 would be filtered off for this one. Every molecule that is less than molecular weight 5,000 would be filtered off. Okay? So, why would removing salts be a problem?
So if you're dealing with oligosaccharide and the size of oligosaccharide is really small.
Right, right. Very good, very good. So in essence, whatever Vimal was saying is absolutely correct. But the basis is that if you have to remove salt and the size of the glycans, if the glycan is just about 3 or 4 residues, 5 residues, 6 residues, you're going to end up pretty close to 1,000. Right? You're going to end up pretty close to 1 to 3,000. And we can't really control. The membranes cannot really just say 1,000 is exactly 1,000. You won't really believe. Obviously, this is related to the pore. And the pores typically have their own distribution. So it's usually not 1,000. Many times even 2,000 molecular weight molecules can pass through. Right? 2,000 molecular weight. So I don't know why. We got some new. I checked in the other room, but it's not there. So even 2,000 can pass through. Not only that, if you have 1 molar, let's say sodium chloride, in the solution, right? And if you have, let's say, 100 ml of this particular eluent that you have collected containing your molecule, what is the amount of sodium chloride that is present in 100 ml? 5.8 grams.
Can we use desalting column?
You can use desalting column, absolutely. You can actually use desalting column. The efficiency of those columns is also, it's high, more than 90%, but you still lose if you are collecting, as he said, and which is why he keeps saying that I can give you microgram quantities, right? Milligram quantities of these N-glycans is a luxury, really a luxury. So if you are trying to separate microgram quantities from gram quantities of sodium chloride, you have to do it extensively. Yeah? There are desalting columns that are available which are many times based on PEG, and they have an efficiency of close to 90 to 95%. You still lose 5 to 10% of the sample, followed by what do you do to collect glycans in general, isolation? What do you do? These glycans are in water. So how do you remove water? Lyophilization. Do you get everything back when you lyophilize? Some of it is lost in lyophilization also. Yeah? So you lose 5 to 10% in lyophilization. So every step you lose quite a bit. Indicating that it is really a challenge to prepare these glycans to a very high extent. Okay, so as and when possible, we try to use other techniques such as precipitation, for example. Okay, by the way, heparin is typically obtained through precipitation. Obviously, we have truckload of heparin that's available from pigs and things of that sort. All right, so. In essence, this is the method for anion exchange. Obviously, it works for cation exchange. You simply have to have different matrix, the opposite type. Okay, any questions on ion exchange? Yeah? Okay, a technique that has been gaining significant value and usage is the hydrophilic interaction chromatography. And this is a technique that in essence is what most people used to call normal phase chromatography. This is a technique which is very similar to normal phase chromatography. So under normal conditions, the matrix silica or other matrices, polymeric matrices, typically have hydroxy groups, typically have polar groups. These polar groups would then interact with hydroxy groups of the sugars, and you would generate interactions under appropriate conditions. So the appropriate conditions are for interaction of the hydrogen bonding, or likewise a little bit of ion pairing interactions. What is typically done is you start with a very high concentration of the organic solvent. In this particular case, for most part, we use acetonitrile. You can also use methanol. Now these high organic solvents actually would force the interaction, which is why we typically use very high concentration of the organic solvent. So those molecules that can actually interact with the polar medium would interact with the polar medium better, and as the concentration of the organic solvent decreases, you would see resolution based in essence upon competition. So as you decrease the concentration of the organic solvent, what do? We increase the concentration of water. So water then replaces the hydrogen bonding capability of the glycans, and that is how resolution occurs. This is a technique that is gaining in usage quite a bit, especially for NNO glycans, The technique that is typically used for highly sulfated glycans is ion exchange. For most part, is ion exchange. Okay. So here, the eluent is not really close to the assay condition of whatever the protein that you are trying to use. The eluent here is organic, or organic water mixture. That's the difference. And this technique is primarily used for preparative purposes or for really analytic purposes. And we don't obviously have the protein involved here. Reverse phase ion pairing. So reverse phase ion pairing is a technique which in essence is primarily reverse phase. Okay, I forget, or not ion pairing. We'll talk about ion pairing a little later. It's primarily reverse phase. So in here, what is done is that you have your carbohydrate molecule. This carbohydrate molecule can be loaded with an appropriate hydrophobic group. Okay, a number of times we can introduce that, or a number of times the carbohydrate itself has a hydrophobic group. He didn't talk about molecules because he ran out of time, but there are, for example, glycolipids, right? Glycolipids are molecules that have hydrophobic group. So for glycolipids, one of the techniques that is typically used is the reverse phase. And the idea is that we utilize the hydrophobic interaction to really resolve the mixture of samples. So these samples would then bind through hydrophobic interactions, and as we increase the concentration of the organic solvent, you keep replacing by increasing the concentration of the organic solvent, we competitively reduce the interaction between the hydrophobic molecule, hydrophobic part of the molecule and the matrix, and thereby resolve the mixture of hydrophobic or hydrophobic group containing glycans. Okay. For most part, glycans are fairly polar, and therefore typically if you simply take glycans just by themselves with so many different hydrophobic groups, this technique is typically not useful. Okay, so keep that in mind. A better technique obviously is hydrophilic. And then we also have reversed phased ion pairing chromatography, which I think I have a slide that I'll cover. But that is also based on reverse phase, except that you can use for highly charged molecules, we can use reverse phase ion pairing. And that technique is very useful, and these days gaining in usage, especially because reverse phase ion pairing coupled with mass spectrometry is a very powerful method to sequence sugars. And in one shot, you also get the structure. So it's very useful. And obviously, there is a technique, hydrophobic interaction, primarily based on hydrophobic groups. This technique can be utilized for sugars, but has not really been utilized for sugars. The idea here is that you might have certain hydrophobic groups in sugars. For example, when we introduce labels that are hydrophobic on sugars to help detect, and I'm going to show you an example, we can actually have those groups bind together, especially with the matrix. And what is done is that by using a very high, by using very high concentration of the salt, you actually force the molecules together to bind through hydrophobic interaction. Okay, so it's exactly opposite. Okay, so this is opposite of what conventional wisdom is, but that's a very nice exploitation of the technique. By using high salt concentration to begin with, what do? We have very high ionic strength of the medium, so all those groups that do not like high ionic strength would be forced together. Okay, and you engineer whatever weak hydrophobic character in these glycans, you engineer better usage of those hydrophobic groups. So they would bind to the matrix, and as you lower the concentration of the salt, then you would then release, according to the hydrophobicity of the molecule, you would then release all different sugars, resulting in separation. Obviously, sugars are very highly polar. There are very, very few groups that can really have hydrophobic character, so this has limited application. All other techniques have fairly good application. Okay, any questions? So these are the, in essence, in a nutshell, very easy to understand principles. Any questions? Okay, good. All right, so obviously there are several challenges to sugar resolution. One of the challenge obviously is something that we have talked about, that when you have a polymeric sugar with these kinds of functional groups, there is, in essence, very little detectability. By the way, you can use not IR, but you can use refractive index as one of the techniques for detection. Okay, refractive index is a change in how light's refractive index, medium's refractive index changes as you change the composition of whatever the buffer is. And when you have these kinds of polymeric species that pass through, the refractive index significantly changes. So you can use refractive index, which is called not IR but RI. And you can use it. Refractive index is independent of molecules, but very few people really use refractive index. But in the extreme case, you can't label it. In the extreme case, you can't label it. There is no chromophore. There is no background chromophore. Okay? And you're left with nothing. You might want to try about RI. Go ahead.
So they do scatter light?
They do, yeah.
They do.
Yeah.
So if you have enough heparin in solution,
the detectability, by the way, yeah, absolutely. The detectability is not very great, but you can still use it, sure. With heparin, you don't even have to go to RI. Unlabeled heparin, you will typically be able to detect easily at 206 to 210. You can. Obviously, if you label it, then the detectability increases dramatically. So, but you can still do it. In fact, I have a slide. Low molecular weight heparin we detected at 206 or something like that. All right. Any questions? Okay. All right. So in order to help with detection, we can have intrinsic chromophore, and this is what I was talking about. For example, this is from a paper that I published a long time ago, where we were trying to characterize these. We were trying to identify the structure of a hexasaccharide that we had obtained. And this hexasaccharide was released by using an enzyme, which you all know of. And this enzyme basically cleaves the interglycosidic bond, generates the double bond between the 4 and the 5 position. And this double bond is in conjugation with that carbonyl group. So this is an alpha-beta unsaturated double bond. And this kind of a molecule, which heparin lyase can typically generate, would absorb to a significant extent. Once again, the extension coefficient of this is not really that great, but enough to be able that we can use it for monitoring reactions, for doing isolation. This absorbs at approximately 230, 232 nanometer. And therefore, we have now generated an intrinsic chromophore which can easily be used in monitoring the digestion so that oligosaccharides can be formed. The other technique, which is most commonly used, is this kind of a labeling. Okay, and this is the reducing and labeling, and we did touch upon this later on. But there are a large number of chromophores and fluorophores available that we can use to label at the reducing terminus. So what is typically done, let's try this one. So what is typically done is the following. So every saccharide typically would have, wow, this is good. All right, this is something that we studied yesterday. Let's say that we have something like that. Or any version of this. So this is always in equilibrium with what we discussed yesterday, right? And that aldehyde is the reactive functional group. So typically reacted with molecules that in essence have structures of this sort. For most part, you can do it with any amine, but if you really want to detect it, if you really want to ease the detection, then you use some kind of a conjugated amine, and there are a large number of groups. Just to give you an example, let's see whether I have a listing. Okay, I don't have a listing, but there are close to 20 different amines that are reported with varying extension coefficients. The commonest one is 2AP or 2N or AMAC. Are we doing an experiment on this today? Yeah. So 2AP is basically 2-amino pyridine, aminoacridone. Where is the amino group? What group are you using? 2, 1, 9, was it? 2 amino. So, and then there are large number of groups, and each of these groups have different detectabilities. They have different places at which they would absorb. So the idea is that you convert that into for example, if you use 2-aminopyridine, you're going to get something like that. That's the step number 1. Typically, this is achieved in the presence of a mild acid. Okay? And what typically happens is that the mild acid coordinates here and enhances the reaction of the amine with the carbonyl group. What is this called? What is this group called?
Shift base.
Shift base, or it's called an amine. If you leave it in water, this can revert back. So this can actually be attacked by water, and hydrolysis can occur. So normally we don't want that, especially, by the way, that technique can also be used and has been used, but I'll talk about that a little later. So normally we don't want such species to revert back, because if they revert back, you're going to lose detection. So what do? Yeah, we reduce it, and we have the most common reducing agent is sodium cyanoborohydride. And sodium cyanoborohydride is going to give you and that's a strong covalent bond that cannot be hydrolyzed. So this cannot be hydrolyzed, and it's stable. And once this is formed, you essentially can detect it very easily. Okay, so that's the basic principle for enhancing the detectability of carbohydrates. So you basically use the reducing terminus, in essence, for most part. Basically use the reducing terminus. Okay, now, I'll just ask the question. I did mention that you can use this technology. You can. What is the problem with this detection system for isolation purposes?
More hydrophilic.
Yeah, this is more hydrophobic. Sure, absolutely. This can be used in a hydrophobic column. Actually, if you leave these molecules, they tend to aggregate, and you would see that they tend to precipitate. So you have to be careful with regard to your glycans. Your glycans are very polar, and at the end you have non-polar. So this is a good combination of if this is polar, this is non-polar. So second molecule will be polar, non-polar. Third molecule, polar, non-polar, and it would form a big aggregate. And if you simply leave it in your refrigerator, you would see that it precipitates out. I don't know how many of you have seen, but that's what typically happens. It arises from that kind of a species. So you'll have to shake it a lot in order to use it for later purposes. But I'm asking a different question. I don't know who answered, but I'm asking a different question. I'm asking a question that this is used for detection, right? We are trying to do this primarily for detection. Can we use this for isolation of glycans? And if we can, is there a problem? Or there is no problem?
You said that's stable, so do you want to revert it back?
Yeah, good point. Very good. This makes it completely stable. So if we are trying to say N-glycan we want to isolate and use it for later purposes, you are going to end up with the terminal residue having modified structure. If you can tolerate that, that would be fine. But if you cannot, then you cannot really get back into the full N-glycan that you want to start with. Right? Yeah? However, this thing, you might be able to convert it back. So if you engineer conditions wherein you allow it to pass through the detector, you detect the molecules, and then you collect the solution and let it hydrolyze, you will get back your for isolation purposes. Yeah?
But the molecule is soluble in water, so...
This is soluble in water.
The carbohydrate usually soluble in water, so what type of solvent we need to use? Because in the...
What type of solvent?
Yeah.
Acetic acid is typically used.
Pardon? Acetic acid?
What are you using today?
DMSO.
DMSO, acetic acid, yeah. That's typically used.
They would still get cleaved, right?
Once you pass it through the detector, you engineer conditions so that it passes through the detector, and then in this particular state, that's what I'm saying. You don't do this reaction. In this particular state, you use it for detection, and then you put it in water, and over a period of time, water would hydrolyze that bond and re-get it. So you can exploit it under really, really tricky conditions if isolation is a problem. So,
Can such compounds form micelles?
Micelles, yeah, they do form aggregates. I don't know whether they can really form those kinds of things.
Bilayers,
I don't know whether they can really form bilayer. There is only hydrophobic group and polar group. It's not positive and negative charge. If you engineer positive and negative charge, maybe they can. So, but this is a common issue. And one of the things is that when we do, let's say, we haven't done electrophoresis, but when we do electrophoresis, the sample is typically sitting for most part. I don't know how many of you have done capillary electrophoresis? You have?
very long ago. I wrote it.
Long time ago. But at that time you weren't working with these molecules, right? But what typically happens is that in capillary electrophoresis, you have your sample sitting inside, it's very small volume, and it's undisturbed, and it's so highly reproducible that, and besides you can store it at a certain temperature, that you tend to leave the sample overnight. Next day you come, you run it, and then suddenly you got beautiful peak yesterday, but today there's absolutely nothing, or very little intensity. And then you keep thinking about, oh my gosh, something happened, but you don't really look at the sample. What you find is heavy precipitation. It's all settled down below. So it's a very common problem. Okay, so all right, so another way that people have used, and this is in the 1980s, 90s, that pulsed amperometry was developed, especially for neutral glycans, and it still continues to be used today, but the usage has declined. So in here we obviously have a pulse. Oops. We obviously have a pulse, and what is typically done, amperometry essentially implies you apply voltage. So that on the detector side, you have a heavy voltage. So there is a pulse of a small voltage and a high voltage. The small short voltage basically oxidizes the carbohydrate so that it generates a current. So these are for most part neutral carbohydrates. So it oxidizes and it generates a current. And this current, generation of this current, indicates the presence of a carbohydrate. Okay, it doesn't say this particular carbohydrate. Obviously, you can use the retention time to be able to identify what carbohydrate or what glycan is actually coming out. But it simply generates a current. And then some of the carbohydrate would basically stick to the electrode. And over a period of time, if you don't clean it appropriately, then the signal would deteriorate and you would see basically no signal. So what is done in every cycle is that you apply a high potential so that you further oxidize and clean the surface. So these are the two cycles, and basically all of this is done fast, so you won't really notice it. So within a fraction of a second you will be able to generate a signal. Okay, most of the time carbohydrate takes several seconds to elute from the column, and therefore this technique is not really a problem. The beauty about it is that you can actually use underivatized carbohydrate. No need to really label. And that's the major advantage. Okay. Now, you have to remember that you don't oxidize all of the carbohydrate. You simply oxidize a very small fraction. So you really lose a small amount of carbohydrate. You don't really lose a whole amount of the carbohydrate. And basically you get 99% of the carbohydrate you can isolate. The pH is typically 12 for most part. Carbohydrates are stable for most part. If there are certain carbohydrates that degrade, then obviously this technique would not be useful. And the final major detection technique in use today is mass spectrometry. There are several ionization protocols. This is absolutely useful for structure elucidation, and with the advent of tandem MS, you can actually do sequencing of every peak that is eluting. Okay, affinity chromatography, just to give you a little bit more idea on terms of the different protocols that you can use to attach the ligand. Okay, and we have several different matrices that are available these days. Okay, you can have a ligand with any carbohydrate with, for example, an NH2 group attached to it. So you can actually engineer a hydrazine as Kubi presented, and you can couple the neutral carbohydrate with a carboxylic acid containing ligand, or for amines, for example, glucosamines or galactosamines, you can actually use it directly for coupling. Obviously, if you're using receptors, then for most part they already have a nitrogen, therefore that's not a problem. Same technology can be used for attaching the ligand to the matrix using carbodiimide. You can use shift-base formation followed by reduction, very similar to what we discussed here. And likewise, you can also use the amines with the aldehyde. So these are all the different matrices that are commercially available and can be very easily used. Thiol can also be very easily used. And as well as you can use practically any nucleophile on the ligand. Once again, that can be nitrogen, that can be thiol, that can be several different receptors of the ligands. Most common affinity-based columns are concanavalin A-based lectin. So this is a lectin, and it typically binds mannose and glucose. So here it binds mannose and glucose, and what we do is we elute with mannoside, alpha-methyl mannoside, and release the there are several other lectins that are available. Basically, you can attach any lectin. And likewise, affinity columns can also be heparin-based, as I said. Heparin columns are actually commercially available. Heparin saccharose is actually commercially available. But you can also prepare it in the lab, especially if you're going to do quantitative, especially if you're going to do preparative chromatography. It's better to use self-made heparin column. Okay. All right, we talked a lot about this, so we can skip ahead. It's already 11:30. So yeah, so this is something that I talked about. This is size exclusion chromatography of low molecular weight heparins, and the only difference that I used was a combination of columns, size exclusion columns. These are underivatized low molecular weight heparins. So you actually detect at 206, and these kinds of nice chromatograms you can fairly easily obtain. Some of these are commercially available. For example, enoxaparin is commercially available. Fragment is commercially available these days in United States. And you can see that, and these are obtained through different techniques. And the idea behind this paper was that each low molecular weight heparins are considerably different. And one of the ways to figure out whether there are, and you might think that this is the same as this, you can probably find there is a difference here, as well as these two are probably similar. But if you really look at number average molecular weight and weight average molecular weight, and chain compositions, then you end up with significant differences across the board. So the idea is to be able to do quantitative. SEC is typically very useful for quantitative identification of number average and weight average molecular weight. In fact, these are the numbers that are specified in the document that FDA utilizes to assess consistent quality across the board. The number average and the weight average molecular weights. And the number average and weight average molecular weights are a reasonable representation of the composition. Okay. Do you know what is number? Yeah, go ahead.
What standard do you use to find these?
Standard?
Yeah, in order to find the actual molecular weights.
RD. And what is it? What standard?
So,
oh, what standards were used? Okay. In this particular case, we used predefined standards, calibration with partially depolymerized heparins. So this is something that you do on a regular basis. Here is the oligo mix, right? And the oligo mix is obtained from depolymerization using heparan lyase. So in here what is done is, for example, this is the peak that corresponds to the disaccharide. Okay, so this is a reference. This is the peak that corresponds to disaccharide. This is tetrasaccharide. This is hexasaccharide, octasaccharide, decasaccharide, dodeca, and likewise. I should have expanded it, but it's fairly clear that it actually goes like that. And therefore you can actually identify. So all disaccharides would appear somewhere here. And I think I used the average for different differentially sulfated molecular weight for this. And the retention time arises from here. And that's how the whole thing is calibrated. Okay? So you had to have a reason. Yeah, good.
Explain MN and MW.
What is Mn and Mw? So Mn is the number average molecular weight. So obviously the formula is written here, but I'll give you a simple.
Can you give a physical sign?
Yeah, I'll give you a physical, and I think for you all it's important to understand the physical basis as to what this is. So number average molecular weight is in essence the A here stands for absorbance, but in actual sense A here corresponds to the number of chains. So we are saying number of chains equals to absorbance. There's a direct correlation. But number of chains times the molecular weight. Okay, so if there is one chain I has a molecular weight of Mi, then you add up all of those different chains and find that and divide it by the total number of chains. So if you really look at number times molecular weight essentially gives you mass of all species, total mass of all species, okay? Total mass of all species. So divided by number of chains. So it's the total mass divided by the number of chains, okay? Physical picture would be if we have all of you, and I know all of you are essentially in the same weight range, but if you had all different weights, then what would you do? Somebody who was 100 pound plus 200 pound, let's say there are 5 people with 100 pounds, 2 people with 200 pounds, and 1 person with 30 pounds. Okay, so 5 and 2, 7 and 8. So there are 8 people. So 5 times 100, 500, plus 2 times, what did I say, 200. So that's 400. So 800 and 1200 divided by 30. So 1230 divided by total number 8. So 8, maybe around, it's the average mass. In essence, number average molecular weight is the average mass of a group. Got it? Now, I'll give you another example to understand what is the weight average. Where is it? Okay, so this is wrong. It should be MW. The equation is correct, but it should be MW. So what is the weight average? So the weight average is the following. So the weight average by, if you really look at this, what is this? Mi times AI Mi divided by sigma AI Mi. Okay? Sigma AI Mi is this term, and Mi times AI Mi is also this term. Okay? So this is simply multiplied by 1 additional mass. So the difference is that let's say that out of these 8 people that we considered, one other person had a weight of 600 pounds. Okay? One person had a weight of 600 pounds. And the remaining 7 had a weight of 30 pounds, 50 pounds, 20 pounds, something like that. Okay? If we do the average, what average are we going to get? We are going to get an average of somewhere 600 plus, if the total here is let's say 100 or 120 or 200, what average are we going to get? We are going to get an average skewed on one side, right? Because this one number is pretty huge. Does that really represent the composition? It doesn't represent the composition. Okay? So what do? We take the proportion of this over the total and do a mass calculation so that we get a more accurate representation of what is the proportion of the average species is a more accurate representation of the weight average. So in general, the weight average molecular weight more represents individual species. Okay? And therefore for most part, number average molecular weight is the one that should be considered, especially if we have skew on one side. How do we know whether we have skew on one side? We know whether by the term called polydispersity. What is polydispersity? It is simply a ratio of MW divided by Mn. So it's higher the polydispersity, greater the inequality amongst the chains. Okay? You have higher the polydispersity, it's more polydispersed. That's why we say heparin is significantly polydispersed. If MW and Mn are the same, for example, if every chain, let's say we have fondaparinux, right? Every chain has exactly 5 residues. Yeah, 5 residues with exactly the same structure. If we do this calculation, you would find that MW will exactly match Mn, polydispersity of 1. All homogeneous species would have a polydispersity of 1. But as soon as we generate heterogeneous species, some chains are big, some chains are small. If you have, for example, here, if you have disaccharide with this species, which is, a huge thing, you have considerable polydispersity. What do I say? Oh, this is not listed here because this is the reference. But in essence, you have considerable polydispersity here. So for such samples, MW is a more accurate representation of the average composition that should be considered in all interaction analysis. Okay?
Basically, it's a weighted average.
It's a weighted average, absolutely. It's a weighted average. Okay? Yeah, go ahead.
So have you seen many, or I guess, is it common to use light scattering to measure polydispersity?
You can use light scattering to measure polydispersity. The only difference is that the particles have to be significantly large in order to scatter light. Okay? So if you can, yeah, absolutely.
Polymers is okay, but not oligomers.
Yeah, oligomers you won't be able to see anything. Yeah.
So Umesh, when you're buying oligos commercially, do they do this kind of an analysis when you go longer than a
Dodecasaccharide.
Yeah.
Well, I can tell you that
do they even
one of my students I was doing capillary electrophoresis of low molecular weight heparins in the presence of different amine containing molecules. Okay, so we have a fingerprinting profile that we had developed. It's a very useful technique to characterize low molecular weight heparins. Now in those fingerprints, what we wanted to do is that how about characterizing each peak using the fingerprint? So can you do that? So we purchased all these oligos that you guys also purchased. And believe it or not, the octosaccharide is not big, by the way. The octosaccharide is a mixture of 4 different molecules. Okay? Now it turns out that the octosaccharide, for most part, out of those 4 molecules, 3 molecules are octosaccharide. But one of them is actually a hexasaccharide. So if you start with an octosaccharide, you already have this thing, you are going to see a lot of higher and differentially sulfated species in decasaccharide, dodecasaccharide, tetradecasaccharide, hexadecasaccharide. I don't really know, you can demand actually, you can, but I don't really know that they do this on a routine basis. But it's not a difficult thing to do. This is not a difficult thing. You have to have appropriate setup, and this calculation is very easy. It looks like a phenomenally challenging task, but that's not the case. Once you set up an Excel spreadsheet, okay, I mean, it's not difficult. I mean, come on, it's not difficult. What do? We basically take on the y-axis, not shown here, but on the y-axis, what is the y-axis here? It is absorbance, right? Absorbance versus what is the x-axis? time. Now absorbance obviously is just direct absorbance. But what is time? Time is converted into, using this, time is converted into mass. So once time is converted into mass, basically, so I don't really remember how many data points we had, but look at this, slice into 1,000 to 100 to 1,000 peaks. So basically you take all of this and you slice it into 1,000 peaks. So you have 1,000 X and Y data points. So the Excel spreadsheet is about 1,000 rows.
Versus time, is it linear?
What was that?
The mass versus time, is that?
No, it may not be linear. That's the reason you have to use calibration. For most part, as I said, this is SEC. So it's a log relationship, actually semi-log relationship that we typically use. Yeah? Any questions? I really wanted to share the paper with you. Okay, what's new here? The only thing that is new here is strong anion exchange versus weak anion exchange. Strong anion exchange essentially means that the matrix has a positive charge at all pHs. Okay, has a positive charge. So this is quaternized, has a positive charge at all pH. Weak anion exchange essentially means that this positive charge can be varied with pH. And strong and weak, for those of you who are not familiar with this, does not refer to matrix analyte interaction. It's not that certain molecules are going to bind strong with the matrix and certain molecules are going to bind weak with the matrix. That's not the case. It actually relates to the pKa characteristics of the matrix or the group that is present in the matrix. Okay? And I think rest of the things we have already discussed. So maybe all right, so this is once again from well, I don't cite the reference, but this is my work, very old, which is the reason this thing is not good. But you can see, some of you were asking me how complex and a profile looks like upon heparin lyase digestion, and this is what it is. See that? This is in essence heparin lyase 1. Only one enzyme has been taken, and we have depolymerized heparin. I don't exactly remember the conditions, but in essence you can see that this is the strong anion exchange column. This is the mono Q column. So in essence you can see that this is one peak and there are several peaks. Each of these, and I had used two-dimensional NMR to characterize, I don't remember one of these. Okay, but we got about 6 really clean hexasaccharides for most part, and we determined the exact structure. Most highly sulfated species, obviously these are really the long things. So in here you can see thousands of species present. Okay, and that's the level of complexity. So when we start, actually when somebody says that I want this particular hexasaccharide or octosaccharide to be prepared from heparin, you have to really isolate it from here, and therein lies the challenge. Okay, it's not really easy thing to do. All right, let's go forward, and you can stop me at any point if you think that you don't understand. So this, for example, is weak anion exchange. I have taken this from Methods in Molecular Biology. This is a very good paper, recently published, and you can actually see that if you couple it, this is pulsed amperometric detection, PAD. HP simply stands for high pressure, and basically they isolated what is it? N-linked oligosaccharides from alpha-1 acid glycoprotein were separated on gradient neutral oligosaccharides. And by using mass spectrometry, you can actually directly identify that this corresponds to that particular structure. Is the power of the technique. Okay. All right, let's skip this. We already did quite a bit. And this is a sample of hydrophilic interaction technique, N and O glycans. using a column meant for hydrophobic interaction, hydrophilic interaction. 2AB is 2-amino benzamide, labeling at the carboxy terminus. This is, what sample is it? This sample is IgG somewhere. Sample 50 microgram per milliliter of fetuin. It's 50 micrograms. And you can actually identify several peaks, and most of these have been characterized, once again, using mass spectrometry. All right, here is a correlation between HPLC and UPLC. How many of you know of UPLC? Do you know UPLC? You know, you don't?
I haven't used it.
But you, I mean, I'm saying knowing, not forget about using it. But you know of UPLC, right? I'm not going to ask you questions, come on. So high pressure or high performance, whatever you want to call, versus ultra high. So the pressure dramatically increases. Typically, HPLC, there is a micron dimension for the matrix. The matrix typically has 5 micron size or higher. UPLC is about 1.7 micron diameter particles. So this is really, really fine particles. And because of that, the absorption characteristics increases dramatically. And there is a certain range in which you use it, you get a linear flow rate, and everything else really works well. The pressures here could be somewhere in the region of 2,000 to 5,000 psi. The pressures here are typically 15,000 psi. So in combination of really fine particles and very high pressure, you have phenomenally increased resolution. So the trend these days, as well as, just to give you a dimension, HPLC, I don't know how many of you have seen columns, you might have used it, is approximately this much. You typically use 15 centimeter, 20 centimeter, 24 centimeter columns. UPLC is just about this much. Okay, that's the size of the column. Length, obviously, the pore is really, really much smaller. The separation is much faster, and I know you can't really see this well, but look at the column. So this is HPLC, and HPLC is 3 microns. This is 1.7 microns particle size. And you can see, one, the profile is different. It's fairly similar also, but significantly different. For example, if you see these 3 peaks here versus these 3 peaks here, much sharper baseline result. Likewise, you can actually see that that particular is a duplet, whereas here you can't really even make out. So in general, these days the trend is to use UPLC. That gives much higher resolution. And look at the dextran ladder typically obtained. Let's stop here. I'll just keep going on and on. I think, have you understood the basics? Yeah? Absolutely, the fundamentals. You should be able to understand quickly from what people have done rapidly, the essence of what they have done. I really wanted you all to look at this paper. I'm going to distribute it. Look at this paper. It's a very simple paper, and the reason why I have chosen it is because this particular paper has a comparison of 3 different chromatographic techniques. It's hydrophilic interaction, reversed phase, and porous graphitic carbon chromatography. So graphite, black, carbon black is really used for chromatography. That's the matrix. So why don't you go, let it go on the other side, and I'll pass it on. That particular thing, yeah. So all of them have it, yeah. And she needs, they need 2 actually. Yeah. Okay? So I'll just take 2 minutes to tell you what I want to really, you all to really, if once everybody gets it, to focus on. Okay, so this is a nice paper. What I would like for, if what I would like everybody to read is obviously the experimental section, to in addition to the introduction and all that you have to read. Normally you all skip the experimental section, but I want you to read the experimental section because there are some nice things, and as you read the experimental section, you try to see whether you understand what is happening during the entire thing, why they have put in certain numbers, why they have put in certain solvents and conditions. And if you understand that, you would have understood how to implement chromatography. The other thing that that strikes me is turn to figure 4. Okay, if you turn to figure 4, and you can see that this is a profile of retention time versus the number of hydroxy groups. See that? And look at the profile. I mean, I wish that they had drawn a line. Is there an R square given somewhere? R square of 0.983. They also say that is the same for RP, reverse phase ion pairing chromatography, very high. But this particular profile is very striking. And that tells me that, well, this is a beautiful tool to really do some nice work and a very simple technique. Okay? You might be able to get away without any detection, without any, not detection, without any labeling. Okay? All right. Let's stop here. Any questions? Okay, good.